This is Heliox, where evidence meets empathy. Independent, moderated, timely, deep, gentle, clinical, global, and community conversations about things that matter. Breathe easy. We go deep and lightly surface the big ideas.
Welcome to The Deep Dive, your shortcut to understanding the really complex shifts happening in science and policy. Today, we're charting, well, a pretty revolutionary frontier, human organoid research. We're talking about growing these miniature 3D human organs, mini brains, mini livers, all starting from just a few stem cells in a dish. It's incredible stuff.
And it's way beyond just being a lab curiosity now. This is actually forming the foundation for, well, future pharmacology, future medicine, these new alternative methodologies or
Exams, right?
They're being pushed, actively promoted by regulators like the FDA here, the European Commission over there. Why? Because these organoids, they just offer much better human-specific predictive data compared to traditional models.
Better than animal models, you mean?
Way better. And it's part of this huge strategic shift really driven by the need to phase out mandatory animal testing and find ethically sustainable human models. It's a massive change.
But, okay, the second you can grow something like complex human neural networks or simulate early human development in a lab, well, the ethical stakes just shoot through the roof.
Absolutely. Which brings us to our focus today.
Exactly. That's why this deep dive is zeroing in on China's 2025 Human Organoid Research Ethical Guidelines. Because this is the world's first comprehensive national governance framework specifically for this. We're going to unpack its, well, pioneering three-tiered structure and what it means globally.
That's right. I mean, while international groups like the ISSCR, the International Society for Stem Cell Research, they offer guidance, general principles, but no national body had ever really codified binding standards, especially for what everyone agrees are the three highest risk areas until now.
And the three are?
Okay, first, brain organoids, the big one. Because, I mean, there's a non-zero risk, however small right now, of consciousness or some kind of rudimentary cognition emerging in these models.
Wow.
Second, organoid chimeras. This is where you're mixing human and animal cells. The risk there is blurring species boundaries, especially if human cells could potentially get into the animal's germline, sperm or eggs.
Yeah, that's a serious boundary.
And third, integrated stem cell-based embryo models. ISEMs. These are essentially synthetic embryos made from stem cells, and they raise just fundamental questions about, well, creating human life synthetically.
Okay, let's unpack this. So we start with the structure, this three-tiered governance architecture. Tier 1 lays out the foundational principles, and what really jumped out at me right away is that this isn't just like taking Western bioethics and tweaking it a bit.
No, not at all. It's actually a fascinating philosophical synthesis. The guidelines very deliberately integrate familiar Western bioethical principles, think autonomy, beneficence, but they weave them together with core Eastern Confucian values. And the result. It's a framework that looks fundamentally different from the, let's say, individual-centric models we're more used to seeing in the West. It consistently elevates the collective interest, the community good, over purely individual rights or gains.
Right. And you see that shift clearly in how they define key terms, like beneficence. Here, we usually think of beneficence as doing good for the individual patient or the research participant.
Exactly. But in these guidelines, beneficence is framed primarily around societal welfare, communitarian norms. The focus is broader than the individual.
And fairness, too, you mentioned.
Fairness also gets a different spin. It explicitly calls for proactive efforts to combat technology-driven stigmatization and discrimination. You can sort of see echoes of socialist goals of equity there.
Interesting.
And then there's autonomy. Now, they do adopt robust dynamic consent protocols, which we'll get into. It's quite advanced. But what's notably omitted is any kind of Western-style mandate for things like intellectual property sharing or direct individual profit sharing that might arise from the research.
So let me see if I get this. The guidelines are kind of saying to the donor, look, your cells, your induced pluripotent stem cells, your iPSCs, they might help us cure a disease ravaging this region, potentially save the village, so to speak. But you don't automatically get a slice of the profits if this leads to a billion-dollar drug.
Yeah.
That's a really different cultural perspective we need to understand.
It absolutely is. And let's make that concrete. Think about a research ethics committee, an REC, in China reviewing a proposal. Say it involves creating brain organoids from a specific donor, maybe someone with a rare condition, but the research aims to tackle a really serious regional epidemic that's flaring up.
Okay.
Now, a Western REC might get bogged down in negotiating the donor's potential IP rights or future profit share. The Chinese REC, though, operating under this communitarian beneficence principle, would likely weigh the potential to alleviate that immediate community-wide crisis much more heavily.
So the public good is baked into the decision right from the start.
Precisely. They embed these socio-ethical priorities, the public good, directly into the decision-making calculus. It's not an afterthought.
That's a crucial distinction. Okay, so Tier 1 is the philosophy, the high-level thinking. Tier 2 must be where the rubber meets the road, right? The 8 general requirements. How do they actually operationalize these philosophical ideas in the lab?
Right. This is about operational rigor and standardization. And there are 3 big mandates that stand out. First, they demand specialized RECs.
Ah, okay.
You can't just have a general ethics board review, say, a complex brain organoid study. That RDC must include relevant domain experts like neurobiologists, neuroethicists.
Well, that makes sense. Need the expertise.
Second, mandatory personnel certification. Anyone involved in this research needs state-accredited training, not just in the science, but specifically in the ethics, the safety protocols, and the specialized technical skills needed for organoid work.
So a standard of competence.
Exactly. And third, very strict national-level management and oversight of all human genetic resources, the cells, the data, and the research results. This is about ensuring traceability, compliance, and preventing misuse.
And central to this operational layer, touching back on autonomy, is this requirement for dynamic consent. Now, this sounds like a major departure from the usual one-time sign here and we're good consent form.
It is. It's a huge shift.
But honestly, doesn't that create a huge administrative burden? I mean, aren't they potentially creating a consent bottleneck that could slow down really fast-moving important research?
Well, it certainly adds administrative overhead, there's no question, but the philosophical underpinning is strong. It's about respecting the donor's autonomy, not as a single event, but as a continuous process, especially given how rapidly these technologies and their implications can evolve.
Okay, explain how that works.
Let's use that Parkinson's disease brain organoid example again. The initial consent might cover taking the skin cells, reprogramming them into iPSCs, and maybe the initial differentiation into neural cells.
Right, the basic setup.
But then, subsequent, more sensitive research phases become specific opt-in checkpoints. Before the researchers can, say, conduct electrophysiological recordings to measure neural network activity, which clearly generates sensitive personal health data, they must recontact the donor and get specific authorization for that step.
Ah, so it's tiered consent, mirroring the research stages.
Precisely. And the most stringent reconsent trigger, the highest bar, is reserved for the activity deemed highest risk, any kind of chimeric integration.
Meaning putting the human organoid into an animal.
Exactly. Transplanting those human brain organoids into an animal model for in vivo testing that requires explicit, separate reconsent. This dynamic model aims to keep the research aligned with the donor's ongoing understanding and wishes. It treats autonomy as a dialogue, not a one-off transaction.
Okay, that makes sense, even with the extra work involved. Let's move to the sharp end then. Tier 3, the special provisions. This is where they lay down concrete technical rules for those highest risk areas. We mentioned brain organoids, chimeras, synthetic embryos. This sounds like where it gets really specific and maybe controversial.
This is definitely where they establish the hard boundaries. For brain organoids, the guidelines mandate things like real-time electroencephalogram EEG monitoring during culture.
Like monitoring brain waves in a dish.
Essentially, yes. And they talk about complexity caps, basically, predefined limits on the structural or functional complexity the organoid is allowed to reach, trying to preempt the emergence of consciousness or sophisticated cognition.
But hang on, using EEG, that sounds like a massive technical challenge, doesn't it?
Yeah.
I mean, EEG signals from these tiny organoids are probably noisy, hard to interpret.
That's the core issue right now.
So you could get a false positive, some random electrical spike, and the rule says you have to terminate the experiment, even if it wasn't actual consciousness emerging. You might be killing valuable research based on, well, technical noise, not genuine ethical concern.
You've hit on the key problem. Relying solely on current, relatively rudimentary EEG methods is likely insufficient. The signals are often noisy, prone to false positives, and frankly, we don't have a perfect correlation between specific EEG patterns in organoids and subjective experience because, well, there isn't any yet.
So how do they make this enforceable or meaningful?
What's needed, and what future iterations of these guidelines will likely need to incorporate, is standardization around quantitative compliance thresholds and, crucially, a hybrid multimodal strategy. Meaning, you don't rely just on EEG. You have to mandate cross-validation with other biomarkers. Things like transcriptomic signatures indicating neuronal maturity, looking for specific gene expression patterns, or morphological evidence, can you actually see complex synaptic structures forming? Or functional network synchronization using techniques like calcium imaging alongside EEG.
Okay, so multiple lines of evidence before you hit the brakes.
Exactly. And you mentioned gamma oscillations earlier.
Right. Why that specific frequency? What's special about 60 to 100 hertz?
Well, in neuroscience, sustained, high-power, spatially synchronized gamma oscillations that 60 to 100 hertz range are strongly associated with complex cognitive functions in actual brains. Things like sensory binding, conscious perception, functional network coordination.
Ah, okay.
So the idea, though still debated and needing refinement for organoids, is that detecting that kind of specific, sustained, high-power activity, not just any blip, would be a much more meaningful trigger for intensified ethical review or hitting that complexity cap.
Wow. Okay, so now your research ethics committee members need to understand neuroscience and signal processing.
Yeah.
That raises the bar significantly, but it forces that cross-disciplinary thinking.
It absolutely does.
What about the other two high-risk categories in Tier 3? The chimeras and isums.
For organoid chimeras, the rules are about strict limits on the ratio of human cells allowed within the animal host, especially in the brain, and mandatory detailed behavioral tracking of the animal throughout its life. This is all aimed at preventing significant humanization of the animal's cognition and, crucially, preventing any accidental germline transmission of human cells.
Makes sense. And the synthetic embryos. Isums.
For the integrated stem cell-based embryo models, the ISEMs, there's an explicit absolute ban on uterine implantation, full stop.
Okay. No transferring to a womb.
None. Furthermore, the guidelines mandate that the culture must be terminated upon the formation of the neural tube.
The neural tube. Now, that termination point is really key, isn't it? Because that's generally earlier than the sort of traditional 14-day rule that's been the benchmark in embryo research elsewhere.
Much earlier, typically. The 14-day rule roughly corresponds to the appearance of the primitive streak, which precedes neural tube formation.
So by setting the red line at neural tube formation, they're being significantly more restrictive? But does that mean they're potentially missing out on crucial scientific data about development that research going closer to the 14-day mark might yield?
They are definitely restricting the window for data acquisition compared to the 14-day limit. There's no doubt about that. But the ethical intent is crystal clear. They are drawing a very bright line to preemptively avoid the ethical and ontological risks associated with allowing the development of anything resembling a recognizable rudimentary human nervous system in vitro. So taken together, these Tier 3 provisions enforced by things like potential electrophysiological thresholds, strict cell ratio caps, mandatory behavioral monitoring, and hard morphogenetic checkpoints like the neural tube, they established the world's first national regulatory framework that tries to tackle these very specific, very challenging neuroethical and ontological risks head-on and collectively.
Right. So China's first out of the gate with these kinds of detailed, centralized technical boundaries. How does this whole approach, this framework, stack up against what other major players, the US, Europe, others are doing? They've been wrestling with these same issues for years, too.
Yeah, it creates a really interesting global governance mosaic because the philosophies diverge quite a bit. In the U.S., the approach tends to be more, let's say, patchwork pragmatism. Yeah, oversight is decentralized. You have federal funding rules from agencies like the NIH, but then a lot relies on local institutional review boards, IRBs, at universities and hospitals. And crucially, the private commercial sector faces relatively minimal constraints. This can lead to, well, some regulatory uncertainty, but also potentially faster innovation, for better or worse.
Okay, U.S. is decentralized, pragmatic. The EU, though, is usually seen as much more cautious, more restrictive on biotech.
Generally, yes.
Yeah.
Highly restrictive, you could say. The EU operates much more unified under the umbrella of the human dignity doctrine, which is really enshrined in instruments like the Oviedo Convention.
Human dignity.
And that doctrine often leads the EU to default towards precautionary bans. If a new technology poses a perceived risk to fundamental human dignity, the tendency is to ban it first and ask questions later. That's why you see non-negotiable bans across the EU on things like human germline editing for reproductive purposes.
Yeah.
Very cautious.
And any other models? Australia, maybe?
Australia is interesting. Their National Health and Medical Research Council, the NHMRC, uses a more nuanced model involving tiered licensing and something they call ethical phase gating. Basically, research projects face progressively stricter oversight and require specific ethical clearances as they move into higher risk phases. It's structured, but perhaps less centralized than China's approach.
So comparing them all, China's distinct advantage seems to be this combination of, like, the EU's unified national authority, but with a much more proactive and technically specific mandate to tackle these future-facing gray zone risks head-on.
I think that's a good way to put it. They have systematic centralization, implementing these unified national standards across the board. They use this tiered risk structure we've discussed, which explicitly confronts those ethical gray zones. Consciousness, synthetic life, collectively, not piecemeal. It really is a proactive, almost preemptive governance model.
So China's moving from just following international norms to actively trying to define the standards for these really high-risk emerging biotechnologies.
Exactly, especially in areas like the potential for consciousness and brain organoids and the oversight of synthetic embryo development. It seems pretty clear this framework is positioned to significantly influence the next update of the international ISCR guidelines, which I believe is expected around 2026.
Okay, so it's influential. But what about practicalities? International collaboration is huge in science. What happens when these different ethical systems collide?
That's the critical challenge now. This issue of dual compliance. Any international collaboration, say, between a lab in China and one in the US or Europe, technically has to satisfy the ethical and regulatory requirements of all jurisdictions involved.
Right.
And that immediately creates potential friction. You have China's communitarian priorities focusing on collective welfare, maybe less stringent individual consent for some data reuse, bumping up against the Western emphasis on individual autonomy, strict informed consent, data privacy, maybe even profit-sharing rights.
So just practically, if I'm a U.S. lab wanting to collaborate using Chinese organoid data or maybe cells derived under their system, I might suddenly find myself needing to meet their dynamic consent rules, even if my own institution only required a basic one-time consent form initially.
That is precisely the kind of tension that arises. It could potentially stall collaborations if there isn't a clear way to navigate these differences.
So what's the solution? Just avoid collaboration.
Hopefully not. The likely path forward involves creating structured processes specifically for ethical negotiation and harmonization. This could take a couple of forms. Maybe developing bilateral mutual recognition agreements, or MRAs.
MRA.
Yeah, where the accredited ethics review bodies in two partner countries formally agree to recognize each other's approvals for certain types of research under specific conditions. Or perhaps establishing joint ethical review committees for specific large-scale projects composed of members from all participating countries who would then negotiate and agree on a common ethical protocol for that specific collaboration.
So the goal is to turn a potential regulatory roadblock into a defined process for dialogue and compromise.
Exactly. It requires effort but avoids deadlock.
Okay, so wrapping up our deep dive.
Well, in short, I think China's framework really demonstrates three key advances that are important for global bioethical governance going forward. First, this idea of anticipatory regulation trying to get ahead of the ethical curve. Second, the tiered scrutiny based on risk level. And third, the absolutely essential cross-disciplinary integration it forces, bringing neuroscientists, ethicists, legal scholars, and policymakers together. It really serves as a vital global reference point, whether other countries adopt it wholesale or just react to it.
So what does this all mean for you, listening? Well, these guidelines are setting really crucial ethical boundaries on things that sound like science fiction, miniature human brains, synthetic embryos growing in labs. The guidelines themselves are just the start. The next critical step, the really hard part perhaps, is translating these ethical concepts into concrete, practical, executable, standard operating procedures, SOPs, that researchers can actually follow in the lab day to day.
The implementation challenge.
Right. And this raises a really important, maybe even slightly uncomfortable question for you to think about. In this constant race between accelerating scientific innovation and the effort to build ethical guardrails, could technology itself, specifically things like AI-powered surveillance and monitoring within the lab, ultimately become the main enforcement tool for these boundaries?
That's a provocative thought.
Imagine, could we see AI systems automatically monitoring an ISIM culture, and the moment AI detects the first signs of neural tube formation, it triggers an alert or even potentially automatically terminates the experiment? The detailed technical nature of these Chinese guidelines seems to suggest that this kind of fusion ethical clarity enforced by automated technical compliance might just be the inevitable future of how we regulate the cutting edge of biotechnology, something to definitely keep an eye on. Thanks for listening today. 4 recurring narratives underlie every episode: boundary dissolution, adaptive complexity, embodied knowledge, and quantum-like uncertainty. These aren't just philosophical musings, but frameworks for understanding our modern world. We hope you continue exploring our other podcasts, responding to the content, and checking out our related articles at helioxpodcast.substack.com.